Okay, so we're talking about chapter 9, muscle histology today. And the fancy word histology is study of tissues. So we're looking at the study of muscle tissues. And we use our muscles to make things move. They are dynamic. And we have skeletal muscles that are going to be our main focus. So I'll introduce cardiac muscle and smooth muscle, but the focus of this chapter is really on skeletal muscles and how they work. And so I tend to go a little bit overboard in giving you guys more physiology than you need at this point in time. But if you are concomitantly taking physiology and or you're going to take it next semester, in both cases, it's good to hear this stuff more than one time. There are 3 types of muscles. Our focus, again, will be skeletal muscles. It's voluntary for the most part and makes us move, pulls on our bones. We do have cardiac muscle. It makes your heart pump blood, right? So cardiac muscle is not voluntary. It pumps our blood through our circulatory system, and that's its job. Smooth muscle really pushes things through tubes. Pushes stuff through tubes, really fancy words. You find smooth muscle in arteries and veins and intestines and esophagus and the bronchi of the lungs. So it's anywhere there's a tube. Skeletal muscles are organs. They contain all 4 primary tissue types. And when we look at just the kind of unique properties of muscle, the number one is it's excitable. So skeletal muscles, or all muscles, can actually shorten. The muscle cell itself doesn't physically get shorter. Well, it does get physically shorter, but no structures are actually getting shorter. What's happening is you have these fibers called actin, and they are going to slide over another kind of fiber called myosin. And so as these slide this way, these Z lines get closer together. And so the overall appearance is the muscles getting shorter, but no structures are getting shorter. It's just structures are sliding past one another. So this is done when it's told to be done, and that's excitability. So the nervous system can release neurotransmitters, releases neurotransmitters, which will be the chemical signal for this to begin happening. Contractility is the ability to shorten, right? So it's kind of like that's what I'm showing you here, and pull on something or squeeze something. Extensibility, now, as I showed you, you can slide a little bit, or you can slide all the way, or you can slide a tiny bit. So there are different lengths. It's extendable over a range of lengths. And the elasticity component is the ability for the muscle to rebound back to its original length after a contraction. So it can return back to its original setup. Functions of skeletal muscles in particular, skeletal muscles pull directly or indirectly on the bones via tendons. That's how they attach. Functions include obviously producing movement. So most of you guys understand that. Maintain body position. So we have something called muscle tone. And this is where your muscles have low level, your postural muscles, your abdominals and back and so forth, have a low level of contraction. It's enough to keep you in your body position, but not enough to actually move you. And this is going on while you're awake. If you're not slumped down on the floor, you have active muscle tone and your postural muscles are contracting, just not moving. You have muscles that support visceral organs, like your pelvic floor muscles. And these muscles will support your organs in your pelvic area. They can regulate the entering and exiting of material, right? So your mouth or your rectum or your urethra. You have skeletal muscle in the opening and at the very end of the line because we have voluntary control over these when things exit and enter. Maintaining body temperature is an interesting one, and it's good to start kind of putting it all together a little bit early in that our body temperature, so some of the energy used for contraction is converted to heat. It's not technically converted to heat. It's lost in the form of heat. So there's energy transfers going on, transferring. And the first transferring is happening when you eat. And so when you eat food, your digestive tract breaks down the food, and it breaks it down into the smallest, monomers like glucose, which is sugar. So I'll use glucose. We could be talking about fats like triglycerides, but the glucose goes into the cell, and you transfer the energy from the glucose molecule, right? So it looks like this. And each of these bonds here, there's energy. And you're transferring that energy, transferring the energy, if you can't read my writing, to a new molecule, transferring energy to a new molecule called ATP, right? And so the energy is now in here. Energy is in ATP molecule. And then your muscle cells are going to use this ATP, right? So you're going to have a second transfer. So the ATP is going to come, and you have this adenosine triphosphate. You have this third phosphate, this high energy phosphate bond, storing energy, and that energy is going to be released to the muscle myofibrils. We'll get to what those are. And that will make the myofibrils slide. So energy transferred to make fibers slide. But not 100% of that energy is transferred. So 100% of energy is never transferred. 60% or so, the energy is transferred. Another 40%, up to 40% of the energy transferred can be lost in the form of heat, right? So energy is lost in the transfer in the form of heat. Now, you guys intuitively know this because when you have your muscle fibers sliding past one another, you are using a tremendous amount of ATP molecules, meaning you're transferring the energy from all these ATP molecules to the little structures that make the myofibrils slide. And so you're using a lot of energy, right? You're transferring it. It's becoming from potential energy in an ATP bond to kinetic energy, actually making fibers slide past one another. But again, 40%, 30 to 40% is lost in the form of heat. And so it makes sense that you get hot when you exercise. And the more you exercise and the more muscle fibers you're using, the hotter you're going to get. And so this heat, whether it's just our postural muscles and it's constantly keeping us at our normal body temperature, or when you exercise, your body temperature can increase, which makes you have to then thermoregulate and sweat some of that heat out. But we use muscles to help us maintain body temperature. And this, also think back when you're cold, people tend to jump up and down, shiver, use their skeletal muscles for this inefficient energy transfer, so they have some extra heat being released. Another thing is when you're really hot, your smooth muscle in your arteries will relax and allow more blood flow out to the skin, taking that excess energy lost in the form of heat out to your skin so you can get rid of it. That's why your skin turns kind of red or pink. You have more blood flow to your skin when you're exercising and hot. And the reverse is true when you're cold. When it's really cold out, those smooth muscles will contract in the arteries and bring more of the blood into your viscerum or your visceral organs, and your skin will actually look a little bit bluer because you've cut down the amount of blood flow going to the skin so you don't lose any heat. So your muscles are very active in maintaining body temperature. All right, so this is a really important diagram to take note of because this is kind of the fundamental layout of a muscle. And so they're kind of showing you like, here's the muscle, right? And then muscles are composed of bundles of muscle fibers, right? And those are muscle fascicles. So muscle fascicles are bundles of cells. We call them fibers instead of cells, and I'll explain that in a little bit. But you have these bundles of muscle fascicles. So there's a bunch of them, and all these are fascicles. And so here's a fascicle, and that fascicle comes out, and the fascicles are composed of a number of muscle fibers, right? So a fascicle is going to have muscle fibers or cells. And so each one of these is a muscle fiber or cell. And within the muscle fiber cell, you see all these little red dots. And those, let's see, what color have I not used? Those little red dots, we're going to make those. I'll use black, even though it's boring. Maybe I won't use black. It's too boring. I'll use orange. No, orange looks too much like this. This is the problem with too many colors. I have too many choices. So these little red dots here, these are called myofibrils. So myofibrils are these red dots. And myofibrils are really the part that slides past one another. The myofibrils have the actin and myosin fibers, or myofibrils that will slide past one another. They're actually called myofilaments. And that's where the actual sliding past one another and shortening happens. So backing up, so you have this muscle, and then the muscle has a number of muscle fascicles that are all bundles of muscle fibers, right? So bundles of muscle fibers, and each muscle fiber has a bunch of myofibrils, right? So there's probably 50 or more in there. And they each have their own connective tissue sheath around them. So a muscle has what's called an epimysium around it. So this connective tissue sheet called an epimysium, that is the connective tissue sheet covering the muscle fiber. And then each bundle or muscle fascicle is going to have a paramysium, right? So paramysium. So this right here is the paramysium surrounding the muscle fascicle, just like I drew here. So each one has a paramysium connective tissue sheet surrounding the muscle fascicles. And then the muscle fiber itself is going to have an endomysium. So endomysiums are these right here, this blue, they go around. So if we look over here, you have the endomysium, it goes around the individual muscle fibers. And then deep to the endomysium, you do have a cell membrane or a sarcolemma. And if you want to see that, that is this membrane on the inside of the endomysium. So here's what makes tendons so incredibly strong, is that tendons are the kind of culmination of the epimysium, all paramysiums, and all endomysiums. So that's a lot of gathering of connective tissue at the ends of the muscles, and it makes them a very, very strong structure. That is why you're more apt to pull the tendon off the bone or pull some of those fibers off the bone rather than actually tear a tendon in half. They're too much connective tissue. They're pretty strong. They're going to kind of start to break down where they interweave themselves with the periosteum on the bone. So you have these muscle fibers wrapped in 3 concentric layers, right? So the first one is the epimysium around the muscle itself. Perimysium surrounds muscle fascicles, and endomysium surround muscle fibers. And so they all interweave at the ends to become the tendons. Tendons, again, connect muscle to bone. You might see the name aponeurosis. It is a tendon that is thick and flattened, and it generally connects a muscle to a muscle. So there's a few aponeuroses that we go over. All right, so the anatomy of a skeletal muscle, so gross anatomy, yeah, there are nerves and blood vessels, right? Because one, they're excitable, so you have to have a nerve attached to it. And where the nerve ending or synaptic terminal makes contact with the muscle fiber, it's termed a neuromuscular junction, right? So that's where the nervous system and the muscle make a connection. And so each muscle fiber is going to have one neuromuscular junction. And so there's some terms that I want to make sure you guys understand, in that on the muscle fiber, where the neuromuscular junction is, you're going to have a motor end plate. So this is on the muscle fiber. And it's where there are receptors for the neurotransmitters to be released. The neuromuscular junction is where the motor neuron kind of connects with the motor end plate on the skeletal muscle. So the neuromuscular junction is the synaptic terminal of the motor neuron and the motor end plate underneath it. And so each skeletal muscle has one neuromuscular junction, and that controls that skeletal muscle fiber. So the axons of the motor, well, the neurons have to go through the epimysium and perimysium, as do the blood vessels, so they can get all the way to the level of the muscle fiber. And then nerves enter the endomysium to innervate each individual muscle fiber. One motor neuron is going to branch. So this is one motor neuron, and they branch to connect to a number of muscle fibers. So this particular motor fiber is going to a muscle fiber there. Here's the neuromuscular junction of a muscle fiber here. Here's one here, and then it's going to go innervate another one there. So one nerve, motor neuron, can innervate several to hundreds of muscle fibers. Now, again, we call them muscle fibers because during development, you have immature muscle cells. Blasts are immature cells, and in this case, they're myoblasts, so that would be muscle, immature muscle cells. And during development, many myoblasts will fuse together and become one long cell. And so since it's a cell that goes end to end, we start calling it a fiber instead of a cell. It's not one cell anymore. It's a number of cells fused together. And each cell brings with it its nucleus. And so these are all nuclei from the myoblasts that have fused together. And so skeletal muscles end up multinucleated. These nucleuses are where you're producing proteins. And these proteins are getting cranked out to make the actin and myosin and the regulatory proteins, and And so there's constant need to create proteins, and so the skeletal muscles are multinucleated to help keep up with the demand. So a couple of terms I've kind of touched on, but we need to define them. Sarcolemma, again, that is the cell membrane of the muscle cell. It is deep to the endomysium. So deep to endomysium. Sarcoplasm is a fancy word for intracellular fluid. Fluid in muscle cell, or the cytosol. And a muscle fiber is the same thing as a muscle cell. And again, it can be very long. It's multinucleated. And these muscle fibers contain myofibrils. And myofibrils contain myofilaments. So we're going to start working on the even deeper levels. It's like Russian dolls, you know, you get a big one and the littler one and the littler one, the little one. We're getting down to the littlest doll, which are the myofibrils. So myofibrils are actually what are responsible for the contraction or the sliding, right? So the tendons of a muscle getting closer together. And each one of these myofibrils is, oops, thought I had something to write on. Each myofibril is surrounded by a sarcoplasmic reticulum. This is a fancy endoplasmic reticulum or smooth endoplasmic reticulum that contains the regulatory molecule calcium. So calcium is the regulatory molecule. And within the myofibrils, surrounded by the sarcoplasmic reticulum, are these myofilaments, and they're arranged in a pattern that we name sarcomeres. Oops. And so we'll get to the sarcomeres. Sarcomeres are made up of myofilaments. So actin, myofilaments are actin and myosin. Okay, so I like this picture because basically it gives you a breakdown. So here you go, here's skeletal muscle, and it's surrounded by an epimysium, contains muscle fascicles. Muscle fascicles surrounded by perimysium contains muscle fibers. Muscle fibers are surrounded by an endomysium and contains myofibrils. Myofibrils, which is this now here, are surrounded by a sarcoplasmic reticulum, which is this. Now, this goes all the way around, right? It goes all the way around. It just doesn't show it in this diagram, but you'll see it in another diagram. They just cut the sarcoplasmic reticulum so you can actually see the myofilaments. So the myofilaments are, the red is actin, and the blue ones are myosin. And thick filaments are the myosin, and the thin are actin. So thick is myosin and the thin is actin. And there's no E. So I'm gonna be so careful because I have it set up to erase everything. All right, so again, this is kind of as you go through sarcomere organization, you know, muscle fascicles consist of muscle fibers consist of myofibrils, and myofibrils consist of sarcomeres. Sarcomeres consist of myofilaments, which are actin and myosin. And so this kind of has you go down the ranking here, and this is a sarcomere. So one sarcomere is from Z line to Z line. But there are a number of sarcomeres. So here's another sarcomere on this side, and another sarcomere over on this side. And then there's going to be another sarcomere. And they just keep attaching, right, all the way down from one tendon to the other. So a myofibril is a series of sarcomeres all attached to each other the entire length of the myofibril. And this is what actually contracts, right? So this slides and shortens. And so with each one of these shortening, the whole muscle just gets shorter, right? So it just keeps getting shorter and shorter. So again, go through these. One thing we haven't talked about is transverse tubules, and I do have a better picture, a little bit better, and you should have a better picture on that if you use the PowerPoint, is that this right here, this is the sarcomere. I'm sorry, Sarcoplasmic Reticulum. So this blue thing, and it goes all the way around a myofibril, right? So they took it off that one, but that one's not showing it, but you can see. And inside the sarcoplasmic reticulum, it contains calcium. And that's a signaling molecule to start the process of actin sliding over myosin. So sarcoplasmic reticulum contains calcium. And so how does your sarcoplasmic reticulum know to release the calcium? Because calcium has to be released out of this tubular structure in order to start the process of contraction. And it knows because these little yellow tubes here along are called transverse tubules. So these yellow tubes are T-tubules or transverse tubules. And that is where the electrical signal flows. So from the neuromuscular junction, you have an electrical signal, and it flows through the T-tubules. So once the electrical signal hits each one of these, it opens up these little voltage gated calcium channels, so calcium can go out into the sarcoplasm. We'll come back to that, don't worry. That's just your first touch on that, okay? So, kind of recapping. Myosin is the thick filament. Actin is the thin filament. Both are arranged in repeated units, and these units are called sarcomeres. Sarcomeres are the smallest functional unit in the muscle. So smallest functional unit in the muscle is the sarcomere. On this diagram, this is a sarcomere from here, and then we don't see the other end of the sarcomere. I'm going to do about here. But it's showing you the thin filaments and the thick filaments. Now, thin filaments slide past the thick filaments. Thick filaments do not move. Again, we'll go back over that. This is showing you, it's not really lining up, but that's a T-tubule. And these are T-tubules. And that's a T-tubule. So that is where the electrical signal, right? So electric signal. I'm really not writing very well. Sorry about that. Maybe it'll make you guys focus a little more on my bad writing. So sarcoplasmic reticulum contains calcium. And this calcium will get released when an electrical signal hits this area. And I believe it's, I don't think I actually discussed it yet. So you have what's called a triad. It's where the terminal cisterna is located and the T-tubule, and when the electrical signal hits that area, that will open up these voltage gated calcium channels in the SR and release calcium. So that is how calcium gets released. So sarcomeres, again, smallest functional unit. of a skeletal muscle fiber. They go from Z line to Z line, and they're in serial all the way down the myofibril. So looking at a closer look at just one sarcomere, you have two Z lines. So you have a Z line here, and Z is for zigzag, right? Little Z's. And you have a Z line here. And so there's that. So this is what defines one sarcomere. Then you have the actin. So the actin are these red, thin strands. And then you have the myosin, which are the thick. Okay, so myosin are the thick strands, and they have these little heads right there. So these myosin heads are popping all up, all around the entire myosin strand. It's made up of several of these myosin tails with heads, and it goes all the way around 360, right? So these little heads are popping up all over the place. So these thick filaments have these heads that can reach across and attach to the actin strand on both sides. And there's a bunch of those heads, so you can have a lot of attachment sites of myosin heads to an actin strand. And the more myosin heads that attach to the actin strand, the more little arms to pull that actin strand, moving the Z lines closer together, because actin is now sliding past myosin. And so each Z line gets closer together, each sarcomere actually gets shorter in length because the actin strands are sliding past the myosin strands. So we're going to talk about that. Now, to return the sarcomere back to its resting length after the calcium or signaling molecule is taken back up, and there's no more myosin heads attached to the actin strand, this little green wiggly stuff here, it's called tintin. It's a type of elastic protein that helps the muscle return to resting length. So the thin filament actin has these different strands. You don't need to memorize the F and G actin strands, but what you do need to understand is on the actin strand, there are some regulatory proteins and binding sites. So this is showing you actin right here. And the ones that you really need to pay attention to are troponin and tropomyosin. So troponin and tropomyosin, these are the basic ones. Troponin, oops, I didn't mean to do that. Troponin binds calcium. So it's a regulatory protein that binds calcium. Tropomyosin is also a regulatory protein that covers up the myosin head binding site. And so when tropomyosin is covering up the binding or active site, then myosin heads can't bind. And so actin and myosin cannot attach, and actin's not going to go anywhere. It's not going to slide anywhere. But when calcium binds to troponin, right, so calcium gets to bind, it's going to move tropomyosin. It's going to move it, right? It's going to roll off the active site. So now a myosin head can attach. Right, so a myosin head will be able to attach when tropomyosin moves off the attachment site. Tropomyosin will only move if calcium is bound to troponin. So that's why these are important to understand. And so when it says, I'm reading this and it's troponin holds tropomyosin in place until calcium binds. Then it releases. Okay, so myosin have these heads, these little myosin heads here. And the myosin heads, are like these little things here. They reach across and they will attach to the active binding site on the actin strand. And when a myosin head reaches across and binds to an actin strand, it's called a cross bridge. So a cross bridge is when a myosin head crosses over and attaches to actin strand. So you can have a couple of cross bridges, meaning a couple of myosin heads attached to an actin strand, or you can have hundreds and thousands of myosin heads form cross bridges. So the number of cross bridges formed is going to be due to how much calcium is being released from the sarcoplasmic reticulum. And so your nervous system has some control over how many cross bridges are formed, and that controls how much tension or how much pull on that actin strand of each sarcomere. So if you have a lot of myosin heads attaching to an actin strand, they're all going to pivot at the same time and they're all going to pull on the actin strand. And that's happening all the way down the myofibril. And so you're going to have a more forceful contraction. So lifting heavy weight, you're going to have your nervous system release more calcium from your sarcoplasmic reticulum to bind up a lot more myosin heads. So you have more heads attached for more strength and pulling. So you have a more forceful contraction. And so important things to understand: myosin heads pivot, but the actual myosin strand or myofilament does not move. Another thing I want to bring up is most of you guys have heard the term fast twitch, slow twitch, right? So fast twitch, slow twitch muscle fibers. This fast or slow is describing how fast or how slow the myosin head can use ATP and pivot. So there's an ATPase enzyme on the myosin head that will hydrolyze and take the energy out of the high energy phosphate bond in ATP and transfer it to the myosin head, so the myosin head can then move. It uses that energy as kinetic energy to make the myosin head pivot, right? And if this enzyme is quick acting and can transfer the energy very quickly to the myosin head, we call it a fast twitch muscle fiber. It has fast myosin ATPase activity, and that myosin head can pivot quickly. It also uses more ATP quickly because it's transferring the energy quickly, and so it moves on to the next ATP. So we tend to run out of ATP with these fast twitch muscles because they're so fast, they burn through their supply of ATP quickly. Slow twitch, that myosin head's a lot slower, so it spares the ATP a little bit more, and it's also, slow twitch is better at producing ATP at a higher rate. So we'll talk about that a little bit later as well. So muscle contraction, it's really, the muscle is shortening in length, meaning from one tendon to another, they're actually getting closer together, the tendons, but nothing's actually getting smaller. It's just the interactions between the myosin thick filaments and the actin thin filaments of the sarcomere. The actin thin filaments are sliding over the myosin thick filaments, and so it's giving the appearance of getting shorter, even though no structures are getting shorter. It's not, I mean, it actually is getting shorter. It's just no structures are getting shorter. And this is triggered by the release of calcium from the SR, Sarcoplasmic Reticulum, which surrounds the myofibrils. And you have to have calcium released from the sarcoplasmic reticulum to have actin slide over myosin, right, to form cross bridges and have those myosin heads move or pivot. And all of it requires ATP. So this is the energy source. Again, you eat food and then your digestive tract breaks that food down to either glucose or fructose or maltose or triglycerides or amino acids for proteins, and it brings it into your bloodstream. And then the nutrients, the amino acids are used to build protein structures in cells, and lipids and glucose or sugar are used to, are really used to extract the energy out of the bonds of a triglyceride or fat molecule, or a glucose molecule, and you transfer the energy out of that molecule and you put it into this new molecule called ATP, and that's what muscle cells use, or all cells use. And so your muscle cells have to continuously produce this ATP if you're using them a lot. And so that is a limiting factor for also fast twitch muscle fibers, as they run out of ATP. This is called the sliding filament theory because actin slides over myosin, right? And so muscle contraction, we start with the neuromuscular junction, right? So this at the neuromuscular junction, right? A nerve impulse, a motor neuron releases neurotransmitter, and I like to just write NT for neurotransmitter. This neurotransmitter is called acetylcholine. You get to learn a little bit about cholinergic neurotransmitters in physiology. And acetylcholine comes in contact with not the sarcoplasmic reticulum, but the motor end plate. I have to say I got this from the book, and I don't like that, so I'm going to cross that out. So acetylcholine binds to receptors on the motor end plate at the neuromuscular junction. Once acetylcholine binds to a receptor on the motor end plate of the muscle fiber, then the muscle fiber forms an action potential that then gets sent down the transverse tubules to each sarcoplasmic reticulum. So this action potential or electrical impulse travels through the T-tubules and hits each sarcoplasmic reticulum of each myofibril in the muscle fiber. So all myofibrils in the muscle fiber are going to get this electrical impulse, and at each sarcoplasmic reticulum, it's going to release calcium into sarcoplasm. And so the sarcoplasm has calcium. Calcium finds its way to troponin on the actin strand. And troponin has a calcium ion bound to it, and that moves the tropomyosin to roll off an exposed active site on the actin molecule, so a myosin head can bind. Okay, so that when a myosin head binds, it forms a cross bridge. One myosin head, one cross bridge, but you can have multiple myosin heads binding. That's going to be dependent on how much calcium ions are released and how much of the tropomyosin has been removed from the active sites. And so once the myosin heads can now bind, they're going to be energized with transferring the energy from an ATP high energy phosphate bond to the head of the myosin. And then once it attaches to the actin strand, that energy is released in the form of kinetic energy. It moves or pivots that myosin head to pull on the actin strand. And so once a cross bridge forms, the myosin head pivots and pulls on the actin strand. So this is kind of a cute little diagram here that summarizes this. So this right here is the neuromuscular junction. And this is the motor end plate, right? And this is a motor neuron. Okay, and the little red dots are acetylcholine. Right, so I'll just mark that for you. That's acetylcholine, neurotransmitter, MT. And so it's released, binds to the receptors on the motor end plate, generates an action potential, and that action potential travels down all the T tubules where it hits this area, the sarcoplasmic reticulum, called the triad. It's going to open voltage-gated calcium channels. Do you need to know this right now? No. Will you at some point if you take physio? Yes. So opens voltage-gated calcium channels. And the reason I'm telling you guys this is to explain something, is that the longer you have neurotransmitter being released, the longer you're going to have the action potential, the longer these voltage-gated calcium channels stay open, the more calcium is released. And the more calcium that's released, calcium in the cytosol, or the cyto, I'll just say cytosol or sarcoplasm, the more calcium will bind to more troponin. And the more troponin that's bound by calcium, the more binding sites will open up for more myosin heads to bind. Okay, so this right here is called a cross bridge. So where the myosin head connects to the, I'll just make that even a more special color. That's a cross bridge. Cross bridge. And so if you look at this, you can have a number of cross bridges. The more cross bridges you have form, the more force or pull. It's like having a tug of war. The more people you have on your side of the rope pulling on the rope, the more force you're going to have to pull that rope. And so the more cross bridges or myosin heads attached, the more forceful the contraction. Okay, so acetylcholine, what happens here is that you have enzymes that break down. So acetylcholesterase, I'm just going to do acetylase. It's an enzyme that breaks down acetylcholine, and so this will stop happening. Right, so this next slide is kind of showing you this. Acetylcholesterase breaks down acetylcholine. At the same time, it's being pumped back into the synaptic terminal. Now there's nothing bound. So there is no signal. No signal, no calcium is being released, and it's active transport back into SR. So you have a calcium pump that actively transports and pumps the calcium back into the sarcoplasmic reticulum. No calcium bound to troponin. Tropomyosin moves back over the active sites, and the myosin head can no longer bind to the actin strand, so the cross bridge is no longer active, and tinting kind of helps the sarcomere return to its resting length. So again, this kind of goes through the sliding filament theory, and that, a couple of things. You have to have ATP bind to the myosin head to pivot the myosin head into its energized state. Then you have to have calcium bound to troponin, so tropomyosin's off the active site so that myosin head can attach to the actin strand, and it releases the energy and pivots the head. Now, a second ATP has to bind again to the myosin head to get it to release from the actin strand and re-energize it so it can attach again and pivot again. So lots of ATP is being used. And so you're going through ATP. At the same time, calcium is being actively pumped back into the SR, so more ATP is used for that. And so muscle cells use a tremendous amount of ATP. That's why they use more energy than most other cells in the body, if they're being used or active. So a lot of ATP is being consumed. And cross-bridging will continue to occur until one of two things happen. The neurotransmitter stops being released from the neuromuscular junction, so there's no more action potential going down the T-tubules. So there's no more calcium being released and calcium gets pumped back. Or you run out of ATP, because if you run out of ATP, you can no longer cross-bridge. And so what happens, with rigor mortis is at death, your muscle cells can't make ATP anymore, and wherever that myosin head was actually cross-bridged at the time of death, it stays attached because you have to have a second ATP bind to release the myosin head. So the muscles kind of stay in that same position. Excuse me. So the amount of tension in an individual muscle fiber. So when we're talking about the amount of tension in a muscle fiber, just one muscle fiber, not a muscle, an individual muscle fiber, is determined by the number of active cross-bridging. And that's going to be determined by the amount of calcium that's released. And the amount of calcium that's released is determined by how much neurotransmitter is released at the neuromuscular junction. And that depends on how many action potentials that motor neuron generates, right? So the nervous system, so it's kind of like the nervous system or your brain forms, I'm going to just call them APs, action potentials, right? Down the motor neuron. And the number generated is going to result in the number of calcium channels open. And the number of calcium channels open determine, how many, or the number of active sites exposed. And the number of active sites exposed determine the number of cross bridges formed. Right? So the nervous system is really controlling how many cross bridges are formed, and the number of cross bridges formed determines the amount of tension in a muscle fiber. Now, the length of the contraction is how long, right? How long is this happening? How long is the brain generating action potentials in the motor neuron? And you're thinking, wait a minute, that doesn't make sense. You said it's the motor neuron that controls the number of action potentials. So when I say number of action potentials, we have another name for that: frequency. So frequency of action potentials. So if it's really, really frequent, you have a lot of action potentials coming one after another. These channels stay open and you have a lot of calcium released. And then you have a lot of active binding sites and you have a lot of cross bridges. You can have less frequent action potentials and you have a little bit of calcium and a few active sites and a few cross bridging. But so frequency of action potential really ultimately determines amount of tension and the number of cross bridging. The length of contraction is just for how long any number of action potentials are being generated. So this is kind of what you get into more when you go into exercise physiology, and that's where, if you're interested in this stuff, it gets fun. Do you need to know this for this anatomy class? Not really. Is it helpful as you go through your education? Yeah, absolutely. So sliding filament theory, this is just kind of showing you, so the A band are the myosin, right? And A, I like to say, is the vowel in dark, right? So the dark band is the A band. And the light band is actin. And the vowel in light is I. So it's the light band. So the I band is the light band, the A band is the dark band. And you see when it's original resting length here, the Z lines are there and there. When it's fully contracted, you notice this light band gets smaller and the Z lines get closer together, but the dark band does not change. And so that's how we know the actin is sliding over the myosin. All right, so kind of just to recap it here in another cute little slide with moons and red dots is that this is the neuromuscular junction. This is the motor end plate. And then again, action potential arrives, releases the acetylcholine, binds to receptors. Acetylcholesterase is there to break it down right away so you can start and stop your contractions quickly. And this is the contraction cycle. So, and they call it excitation contraction cycle. So basically, calcium gets released, right? Binds to troponin here. We have a little better color. Binds to troponin here. Moves tropomyosin. A cross bridge forms. And it's showing you from all different directions, depending on how many calcium molecules are bound and how many tropomyosin are exposed. It's exposing these active sites here. And as soon as the myosin head attaches, it's going to pivot and it's going to pull, right? And so the more cross bridges, so that's a cross bridge, and that's a cross bridge. The more cross bridges are there, the more force of the contraction. So again, number cross bridges formed determine force of contraction. And the number of cross bridges formed is really determined by the frequency of action potentials arriving at neuromuscular junction. So your brain ultimately determines the number of cross bridges formed by your brain deciding how many action potentials are going to be generated on a motor neuron that innervates muscle fibers. And that frequency is going to determine how many cross bridges ultimately form in a muscle fiber, and that will determine the force of the contraction. We will get to how your brain controls varying the number of muscle fibers that it activates, right? So remember, you have a muscle that has bundles of muscle fibers called muscle fascicles, and you can actually vary the number of muscle fibers that are being activated within that muscle. So you don't have to use the whole muscle every time you want to move something. You can activate only some of those muscle fibers, right? And so, and you can activate them with a high amount of frequency, so you have a lot of cross bridges in those particular muscle fibers, but you have a lot of other muscle fibers that are not being activated at all. So you can vary the amount of force of the contraction by varying the number of muscle fibers that are activated, not just the frequency of that activation. So back to this, you need a second ATP to release the myosin head and re-energize it. And this little orange area is where that enzyme is that energizes. And that binding of the second ATP will release the myosin head and re-energize it as long as there's calcium present and tropomyosin is off the active site. So this is kind of the older version. Once stimulation stops, remember, calcium is pumped back into recaptures is a nice way of saying, you know, it's pumped. Active transport back. Right, so that's why they show you this tug of war. It's like myosin heads, cross bridges. I do want to end here, and then I'm going to have a part 2, so we can finish this because this is a rather long portion. So this is just part 1, and we'll start back with this in part 2.